All right, so in this section, in this video, we're going to be focusing on proteins and how proteins relate to cell parts. Some cell parts have proteins as part of their structure, and then we're going to be focusing on the cell parts that have to work together to make the proteins that the cell needs. So first we're going to take a look at the plasma membrane or the cell membrane. Whenever you see a diagram of a cell, that outermost structure there oftentimes is the cell membrane. If you zoom in on the cell membrane, this is really what it looks like. So it's showing you like this little line right here that we label as the cell membrane. If you actually zoom in there, then this is what the cell membrane is made out of. We've talked about them before. Those circles with the little tails are called phospholipids. So the cell membrane is made of phospholipids that are arranged in a bilayer. Now, bi means two. So if you take a look at, you're going to see you got one row of phospholipids, and then you have another row of phospholipids. So that's why the membrane is called a phospholipid bilayer. Now, one thing we'll see is that in the membrane, there can be proteins, and these proteins allow molecules to cross in and out of the cell. So sometimes molecules are going into the cell, sometimes molecules are going out of the cell, and then sometimes molecules can't go either in or out of the cell. So let's just show there's a molecule right here, and let's indicate it can't cross. That relates to the function of the cell membrane. So the job of the cell membrane is to regulate what can go in and out of the cell. So since some things can go through it and some things can't, we can't say that the membrane is permeable, because that means everything can go through it. The membrane is semi-permeable. Let's underline semi-permeable. It just means some things can go through the membrane and other things can't. Now, as I mentioned before, we're going to be focusing on proteins. And even in the cell membrane, there can be proteins. There can be proteins in the membrane. And I showed you that up here in this diagram, and I showed you there's two layers of phospholipids. But you can also see that in those phospholipids, you can have proteins floating around. And those proteins can have different jobs. So down here, I'm just attempting to explain to you that the reason why some cells can do jobs that other ones can't is because of the proteins that they have in their membrane. So this is showing you right here, it's kind of showing you a structure here, and they're trying to demonstrate that this is a protein that is embedded in the membrane. Now this is a cell. The job of this cell is to make melanin. Melanin is what darkens your skin after you've had some exposure to the sun. And if you kind of look at this process that's happening, you can see that this cell itself, it doesn't make the melanin. It can signal, so it receives the sun's rays, and that can cause it to produce a molecule, and that molecule can travel outside and hook onto one of these proteins that's in the membrane of this cell. And it's this cell then that will activate the making of melanin. You notice that this cell down here, it doesn't have that protein. So this cell that's receiving the signals, it can't signal for it to make melanin because this cell doesn't have that same protein in the membrane as this melanin producing cell. Okay, so can this cell send a message to this cell to make melanin? No, it can't. Because in order for a message to be sent, you have to have a protein that can receive that message, and the cell can't receive that message. Does the adipocyte have a gene in it, in its DNA coding for melanin? So this is from last class. You have to remember that all of these cells, if they all are from you, if they're all here in the skin, they are genetically identical. But the reason they look different, the reason why they can do different things, is because they turn on and off different genes, making them have different proteins. So all of these cells, they do have the gene for melanin. Obviously this one does because it turns it on and then the cell makes melanin. But this cell also, it has a gene for melanin, it just keeps it off. And this one also, in its nucleus, it's going to have a gene for melanin. It just never turns it on. So they all are genetically identical. All right. Now, we just saw that cell structures like the membrane can have proteins, and those proteins can do different jobs. Now we're going to focus on a lot about protein production and transport. So we're going to look at a bunch of cell structures that have to interact with each other to make proteins and get them where they need to be. Now, this all summarizes for you what the nucleus does, what ribosomes do, what the roughy are, and what the smoothie or the Golgi does, not the smoothie are. And you guys have already seen some information related to that because you had a video. Let's see if I can find it. It's this one right here. And we watched it several times as we were getting introduced to cell structures. And it talks about how, I'm going to hit play here, and I'm going to narrate it and pause throughout. And this is what you need to be able to tell me about how the cell is able to make different proteins. So here in the nucleus, we have the DNA, and the DNA has sections of it which are called genes. Now, when we go to build a protein, it's going to happen out here in the cytoplasm, outside of the nucleus, and it's going to happen at an organelle which is called the ribosome. Now, our DNA is an important molecule. We don't want anything to happen to it. So instead of just sending the instructions, the DNA, out to our ribosome, we're going to make a copy of it. So in the nucleus, the cell is making a copy of the DNA gene. It's making a copy of the instructions, and basically this copy is going to tell the ribosome how to build a protein. So that's what you see here, is these are copies of a gene, and they just left the nucleus, and they are now in the cytoplasm where there is a little tiny organelle called the ribosome. And the ribosome is clamped down on it, and you can see something starting to be made here, and that's going to be the protein. Now, to help you remember that ribosomes make proteins, we might think about if you're having ribs tonight for dinner, those are the protein in your meal. So if you look at ribosome, the first three letters is rib. So if you remember ribs are proteins. That might help remember that the job of ribosomes is to make proteins for the cell. Now let's watch what's going to happen to this protein when the ribosome is done building it. This ribosome here is called a free ribosome. It's unattached. It's just floating around in the cytoplasm. And when they make proteins, they're just going to release the proteins here in the cytoplasm. And these proteins are now going to do a job in the cytoplasm for the cell. So this protein, it was made because the cell has to put several little pieces of that protein together to make another cell part, which is the cytoskeleton. So if I back up here, this is called the cytoskeleton, and each little part of it here is made of a protein called tubulin that was made by a ribosome. Okay. Now let's take a look at, because I got a different ribosome here, and it received the instructions, the copy of the gene. And by the way, that copy of the gene has a name. This molecule has a name. It's called mRNA. And mRNA is called, or that M stands for messenger. Because if you think about it, messengers take information from one place to another. So back here in the nucleus, when we built this mRNA, this is the instructions. We're going to take those instructions out to the ribosome, and the ribosome can read the instructions and build the protein. Okay, so see, we have free ribosomes, and when they're done, they're going to release those proteins into the cytoplasm to do a job. But then notice that we have some ribosomes that are on this structure, and this structure is called the ER, which is short for endoplasmic reticulum. Now, because this rough ER has ribosomes on the surface, if you look at it under a microscope, it's going to look really rough because it's got just thousands of these little tiny dots, these ribosomes all over it. So it kind of looks almost like sandpaper. You know, paper is smooth, but if you glue little specks of sand to it, it's going to look and feel rough. So that's why it's called the rough ER, if it's got ribosomes on it. Now notice what happened is the ribosome read the information, read the mRNA, and it's building this protein, and it's going to put the protein in the inside of the rough ER. And then that rough ER is going to modify this protein, package it up in a vesicle, and is going to send it to the next cell part, which is the Golgi. So the Golgi is like the post office of the cell. And you can see the Golgi kind of looks like several pancakes stacked. And inside, you're going to see a bunch of different proteins. So the rough ER can send proteins to the Golgi, and then the Golgi is going to tag those proteins to go 3 different places in the cell. So let's watch what's going to happen. So we're going to watch this vesicle come off the Golgi, and let's watch what's going to happen. There's actually 2 proteins there. There was the one that we just saw the ribosome make and put in there. So let's just watch that. Let's watch what happens at protein. Looks like that vesicle just fused with the membrane and became a part of the membrane. And now that protein that was in the middle of the vesicle has been released from the cell. So if you ever hear like your cells secrete or release insulin or adrenaline, those are proteins. And they traveled in a vesicle and eventually got put outside of the cell. Now I'm going to back up a little bit because this Golgi has also sent another protein in this vesicle. So here's that one that we just watched, but let's watch this one that looks kind of like an X. This protein is now stuck in the cell membrane. So if you go back to our notes, and I showed you that there are proteins that are in the cell membrane, now you know how they got there. Is the Golgi can send them, and then they can become a part of this phospholipid bilayer of the cell membrane. All right, now I'm going to back up one more time because we just saw one protein get released, one protein get embedded in the membrane. I'm going to back up and we're going to see what's happening here with this vesicle. So let's go back to about right here. Okay, so this Golgi put some proteins in this vesicle, put some proteins in this one. Let's take a look at what's going to happen here. So this vesicle isn't going to fuse with the membrane. It's going to come over here and it's going to fuse with this organelle, which is a membrane compartment as well. And it now looks like all those proteins just got put in this compartment. So this is a lysosome. A lysosome contains a bunch of digestive enzymes, digestive proteins, and that can help the cell break down unwanted materials, which I'm going to show you here in just a second. But let's recap. So we have ribosomes. Ribosomes are going to get instructions. The instructions are called mRNA. So the mRNA is made in the nucleus, and then it's going to leave the nucleus. It's going to go out. This is a ribosome in the cytoplasm. It's called a free ribosome or unattached. It's going to read the mRNA, the information, and it's going to build a protein. When it's done, it releases the protein right into the cytoplasm. But this ribosome is receiving instructions to build a different protein. That ribosome is attached on the rough ER. It put that protein in the rough ER. The rough ER will modify that protein and send it to the Golgi. The Golgi is going to modify and tag proteins for 3 possible destinations. Some proteins will be tagged to go out, some proteins will be tagged to be embedded in the membrane, and some proteins will be sent to the lysosome so they can do their job. All right, so now that we've seen that animation, what I described to you is what I'm describing to you already on this piece of paper. But let's take a look at this model and see if we can figure out some of the different parts. And look at it. What does it look like as a drawing versus an animation on a video? So hopefully you can recognize that this is a cell. It looks like we have, this would be our nucleus right here. And in the nucleus, we have DNA. So DNA is a really long, stringy molecule, and that's why it's almost drawn like a string. And it's kind of hard to see on this one, but there is a light little molecule that's in here, and then it comes out, and then it's right here. And that would be the copy of this section of DNA. Now, sections of DNA that are the instructions for building proteins are called a gene. So this would be what a gene is. And then maybe further down, there is another gene. And it can end up getting turned on and copied at a different time. Okay, so genes are just sections of DNA. This copy, again, is not DNA, it's called mRNA. And that mRNA is going to come out of the nucleus, and eventually it's going to go, in this case it looks like it went to a ribosome. And this ribosome, since it's on this surface, and this is the rough ER, I'm going to put RER for rough ER. It does have it right here. It's the rough ER. This ribosome, we're going to say is an attached. So let's indicate it's an attached ribosome because it's been attached to the surface of this rough ER. So it's reading the mRNA and it's going to build a protein and release it here in the rough ER. So let's draw some arrows here. So that little dot right there is my attached ribosome, and that little squiggly molecule there is trying to show you that's a protein that is now in the rough ER. And then that rough ER is going to modify and ship out that protein wrapped in a membrane, and that would be what we would call a vesicle. And then that vesicle membrane can just fuse and become membrane of the Golgi, and now that protein is here. And then eventually that Golgi is going to take that protein, tag it, and it put it in a lysosome. So this is saying right here that this is the structure. It's not going to go to the cell membrane. It's a lysosome. And now there's some enzymes inside the lysosome that can digest molecules, which I'll show you here in a second. And let's go over here. And we've got another attached ribosome. Again, it got instructions, mRNA. It built a protein, put it in the rough ER. The rough ER is packaging it up and sending it to the Golgi. And then the Golgi can package it up and send it. In this case, it looks like it is being secreted outside of the cell. Now, sometimes whenever proteins are made, they're not put inside, which is called the lumen. They're embedded in the phospholipid bilayer, in the membrane. That's the outer part of this like rough ER. So then when this piece breaks off, you can see the proteins are in that phospholipid bilayer that is wrapped around that little compartment. And now we have proteins here, and then pretty soon they're packaged up in a vesicle and they're still embedded in the membrane. So when this phospholipid bilayer gets to this membrane, which is a phospholipid bilayer, it just becomes a part of it. So these phospholipids and proteins in this membrane right here are just going to become phospholipids and proteins that are in the cell membrane. Okay, so you can see that those proteins now are embedded in proteins in the membrane, like I showed you, like these. That's how they got there. All right, so again, most of this is just summarized for you right here, but there's 4 places, or let's just start with the 3 places. So attached ribosomes can make proteins, put them in the rough ER. The rough ER can modify them and send them to the Golgi. The Golgi can tag them and send them 3 different destinations. 1, send them to a lysosome that's going to stay inside the cell. 2, put them in a vesicle that'll then eventually fuse and dump them outside of the cell. Or 3, embed some of those proteins in the membrane like you see right here. And that's summarized for you right down here. Okay. Now, before we go on to the next page, I want to talk about how those vesicles get around, because it looks like they just float, but they don't float. They're actually being walked along what's called the cytoskeleton. The cytoskeleton is basically a protein highway. So cytoskeleton crisscrosses all over the cell. So let's add some little beams that crisscross, and let's label these are the cytoskeleton. And in the video, you saw some cytoskeleton. But click back on that. I'm going to fast forward here. So cytoskeleton is made of little proteins called tubulin that have to be assembled into the cytoskeleton. So you can see the cytoskeletons all over the place. It's like highways that crisscross all over the cell. Okay, and so how these vesicles are actually moving is they're being walked by little proteins that looks like they have little legs, and they're being walked along the cytoskeleton to other parts of the cell. So like this one, and actually I'm going to show you more I'm going to make a cytoskeleton kind of run this way. I'm going to show you there's a little protein that looks like it has two little legs and it attaches on and it's walking along the cytoskeleton. I do have a video that shows you more what that looks like. So this is kind of showing you, this is cytoskeleton. This is the beams that crisscross all throughout the cytoplasm. And then this is a vesicle, and inside of it we'll have molecules like proteins. This is that protein that has little legs, and it's, again, it looks like it's walking along that cytoskeleton, and it's taking that vesicle from one side of the cell to the other side of the cell. Now, I forgot to mention why I'm here. I need to go back a little bit. I talked about ribosomes like this right here that's attached, and they make proteins and put them in the RefER. But let's take a look at this little ribosome out here, because this is a free ribosome. So we can say free, or we can say unattached. It's a free or it's an unattached ribosome. And just like these ribosomes out here, mRNA is going to be made and sent, and that ribosome is going to attach on to the mRNA. I'm going to label as mRNA, and it's going to build a protein. And when it builds that protein will break off, fold up, and now we have a protein that's just released here in the cytoplasm, and it can perform different jobs. Like I showed you, some proteins will come together to make cytoskeleton, okay? All right. On this page, I just wanted to show you a few more pictures about like what do these lysosomes do, because I just kind of showed you that how lysosomes get their proteins, how they get their digestive enzymes. So remember a lysosome, so this is a lysosome right here, and lots of times lysosomes, it's showing you that if this breaks off and goes here, that's going to deliver some proteins, some enzymes into this structure, which is lysosome. Lots of times lysosomes is going to look like a chocolate chip cookie. And inside of that, those little dots they put inside, those are digestive. They can break down molecules. So they're digestive enzymes. Remember, enzymes, we learned, are proteins. So the Golgi can send those digestive enzymes to the lysosome. Now let's take a look what the cell's doing. So if you remember, we talked about how your white blood cells can engulf bacteria and bring them in and destroy them. So let's say those are some bacteria cells. You can see that cell membrane wraps around them and traps them in a vesicle. So now we have part of the phospholipids that used to be in the membrane are now around this little compartment. And then if this fuses with this, then those enzymes are going to be able to get in there and break down the bacteria and destroy them. Now, lysosomes also, because I indicate right here, white blood cells, they can trap bacteria, and they can use their lysosomes to destroy the bacteria. But lysosomes also can help digest old, worn out cell parts, like the mitochondria. So here that's showing you this is a mitochondria, but it can become damaged, and the cell needs to have a way to get rid of it, otherwise it's going to fill up with organelles that don't function. Well, see our lysosome here that has those digestive enzymes. So let's make sure you understand that these little circles inside are enzymes in the lysosomes. So they can fuse with this mitochondria, and now those enzymes can break down that mitochondria. Here's what I mentioned about the cytoskeleton, because we're talking about how the cell can make proteins and send these proteins to different parts. We know that proteins can be put in vesicles. So here's a vesicle. And let's just show that there is some proteins inside. So let's indicate that that is a protein. Let's go ahead and name one. Let's name a protein that can be made in cells and then released outside of cells, like insulin. Okay. And so then it's in a vesicle, coming from like the RefER to the Golgi, or from the Golgi to the cell membrane. And then eventually that protein is going to be released outside. And again, the cytoskeleton, they look like, sometimes they zoom in and they look like big tubes. The cytoskeleton. Sometimes they just draw them as kind of skinny lines, and they might show little vesicles being moved along. But that's the job of the cytoskeleton, is it kind of creates these highways where molecules like proteins can be moved from one cell part to the other, okay? And then the cell membrane. We've already talked about the cell membrane is related to proteins. It's got proteins in it, in the phospholipids. It also has proteins, or proteins can be released out of it. But I like this diagram because it's showing you how this can actually happen. So in this vesicle that's being sent to the membrane, this is a protein that's been embedded in those phospholipids. So this right here, this is a protein. So I wanted to show you that proteins can be kind of stuck here and embedded amongst the phospholipids. And when this vesicle gets close to the cell membrane, then there's proteins in the membrane. So that's what they're trying to show you there. They're just kind of drawing them a little bit different than what you maybe have seen in the previous diagram of the cell membrane, but that's a protein. And when these get close together, they get kind of stuck together here. So notice that this clamped down on this. And so then when this vesicle now is attached here to this membrane and to this protein, these phospholipids can kind of intermingle. And you see how then they can just kind of come together, and that makes a little opening here when they get rearranged, and now all that stuff that's on the inside can get released. And these molecules can now go out of the cell. So that's really how all that works. All right. So the last thing I want to relate this to cells, because remember, cells are specialized, is we just taught you how a cell and all of its parts can work together to secrete. So let's put secretion of a protein. So this is a vesicle, this is a bunch of protein that's coming out. That's the job of this cell, is to make a protein and to release it for whatever reason, okay? Let me give an example of cells that do that. So you have your beta cells in your pancreas. This protein that they make and they release, that is actually insulin. If someone has type 2 diabetes, type 1 diabetes, lots of times these cells have died, and so there's no cells to make the insulin and release the insulin. So it's hard for those people to control their blood sugar. So one thing to note, this is an example of a specialized cells. So the cells in your pancreas that make insulin, those are specialized cells. Some unique features about them is they're going to have really large amounts of these cell parts that are needed to make that protein insulin. They're going to have lots of ribosomes, a lot of rough ER. There's going to be lots of Golgi, even more than what we see right here. But you're going to see it packed. So if you want to emphasize that and put even more like rough ER and even more Golgi, the whole cell is just packed with these protein producing and trafficking organelles that can then release all of these proteins and vesicles that can go to the membrane and be released, okay? So you're going to see that that cell is going to have vesicles just full of insulin. You're not going to see a skin cell look like this because skin cells don't make insulin. Another example of a specialized cells is a goblet cell. And goblet cells are with those other cells that have cilia. Remember, the ciliated cells move the mucus, while the goblet cells, they make the mucus. They make mucin. Mucin is a protein. If you mix mucin with water, you got mucus, okay? So these goblet cells, they're going to need a lot of those cell parts if it's their job to release a bunch of mucus or mucin into the exterior of the cell. So it can that mucus can trap particles that you're breathing in. So that's another specialized cell. It's called a goblet cell. You might think of I think of a goblin, and usually like a goblin is like kind of drooling and salivating and kind of mucusy. It might help you remember like goblet cells are the types of cells that make mucus in your lungs. And so again, these cells are going to have a whole bunch of ribosomes attached onto the rough ER, so they can make just millions of molecules of mucin every hour. And they need to send them to the rough ER, and then the rough ER is going to send them to a lot of Golgi, and so those cells are going to have a lot of vesicles as well, waiting to release all this mucin into the outside of the cell. think that's it for our cell parts that interact to make proteins.